Well, let's see if it's possible to get a sort of overview of Module 8, because there were quite some number of topics covered here. So I have tried to make a sort of short overview. And it's all about comparative genomics in this module. Comparative genomics. And what does it mean? It means we want to compare genomes to each other. And which genomes are we talking about? Well, it's usually some genomes that we have sequenced ourselves and then assembled and put together, like we have done now for several weeks. But it could also be genomes we have downloaded from some databases that other people have sequenced. Right? So it's quite often a combination of these two. Right? We have sequenced some genomes and we want to, well, we want to compare them to each other, but we also want to put them into a bigger picture, right? What have we seen before when it comes to similar genomes? So it means in any case, we have sort of many genomes now, right? Not one, like we have more or less done up to now. Now we have many. And the reason I stress this is that some of the things we do in Module 8, of the more technical things we do in Module 8, is, I mean, this arrow job and looping and so on. This comes from the fact that we now have many genomes. Why do we want to compare genomes? Well, this is actually a big thing and not really, as I said at the very beginning of this course, this is about, in BIN310, we are mostly concerned about how to compute and not so much why we do these computations, because there are many different reasons for that. But basically, we can say that we want to see how the genomes are related. This is a very common sort of task. How are the different genomes related? And why is this important? Well, this is important for instance, if you have an outbreak of a pathogen in a hospital, that's a classical example. Outbreak of some pathogen, right? People get ill. Then you want to see where did this come from? There are probably some routines that have been going wrong, right? About hygiene and something. So in order to find out how could this pathogen get into our hospital, it's often important to be able to compare this pathogen to other similar pathogens we have, or where have we seen something similar before, right? So that's why this question is often asked. And the example I have in the introduction is from the pandemic, right? That's really a good example. We want to understand how this SARS-CoV-2 virus, where did it come from, right? That's been one of the big questions here. We know it sort of popped up in Wuhan in China, but actually where does it come from? There are also some other reasons for comparing genomes, and that so notice that this has to do with evolutionary history, right? This has to do with evolutionary history, relatedness. But this other one here, some of the genomes have some special genomic features? That's a slightly different question, because now we don't really ask how they are related, but if they have some properties in common, right? Do some of these genomes share some properties? This is not something we will focus on now. I just mentioned it, so I sort of almost wipe it out again, because we are focusing on the relatedness here now. But I mentioned this because this is also important in many aspects of comparative genomics, and this leads to what we know as Genome-Wide Association Studies, GWAS, you may have heard of. But we will not dive into that here this year in BIN310, but perhaps some other year. Many of these applications, when we look for relatedness between genomes, they require high resolution. And what do I mean by that? It means that we want to see how one particular strain relate to the other strains within the same species. So we are not interested in comparing genomes across various species and at a big evolutionary picture. We are only looking at some very sort of narrow part of the bacterial biology. So for instance, within one species, right? We know, for instance, that E. coli, most E. colis are harmless, but some of them are really dangerous. And it doesn't help us to compare these dangerous E. colis to all kinds of other bacteria. We want to compare them to other E. colis, right? Same species. And this means we are actually interested in the very recent evolution. So when we talk about relatedness here, we talk about what has happened very recently. We are not interested in the long-term evolutionary history of these organisms for these applications. For other applications we may, but not for these. If you want to investigate the outbreak of a pathogen in a hospital or in a food production pipeline, it's not interesting to see how these organisms were related to other organisms 10,000 years ago. We want to see how they were related last week, right? Or a month ago or something like that. Short time history. And how do we actually display relatedness? Phylogenetic trees. So that's what we are doing in Module 8. We are plotting phylogenetic trees because that's the way we display relatedness. So the first thing we do in Module 8 is to use the classical approach of a marker gene comparison. That's the classical way. And this is this means we extract the same gene from each genome. So instead of using the whole genome, we only focus on one single gene. But the same gene is taken from each of the genomes. Then we align this using some multiple sequence alignment tool. And then we build a tree from this alignment. And this is more or less exactly what we did in BIN310. So I recommend you to have a look at week 8 and 9 there if you want to repeat this, because this should actually be known. How do we make phylogenetic trees from a single gene? Standard approach. And of course, in this Module 8, we use the 16S gene. It's not like you have to use the 16S gene because there are usually other genes you can use as well, and we will see later. But this is at least the very most commonly used marker gene for comparing bacteria and also archaea. All genomes should have such a gene. That's the first advantage of using this one, because you are sure to find it in all genomes. However, be aware that when you do sequence assembly yourself, and we have done so, this 16S gene is not always found among the contigs. Not always found among the contigs, and that's a problem really. And why is this? That's strange, right? Why wouldn't this gene be in the contigs? The reason is this particular gene is one of the repeated regions. We have learned that the major obstacle, you could say, for getting a good assembly, at least for short read data, is the fact that we have repeated regions in our genomes. And this one is one of these. Most genomes have this gene in multiple copies, and they are more or less identical to each other, right? So be aware that the 16S gene may not always be present in the contigs. It's surely present in the genome, but due to the assembly software, you could say, and sequencing with short reads, there may be some problem getting hold of it. So the problem with the 16S is, however, that this is so highly conserved that it will only reflect the long-term evolution. The long-term evolution. So it's not really suited for this short-term history that we are usually or very often looking for here. I should actually, before we move on, have a look at this section 3 in module 8. So what that we do actually this thing. We used a software barn up here to actually extract this gene from the genomes, and it turned out to be difficult to do it in this way by running it from R, because yeah, you can read on the discussion forum. But anyway, you can see we need to loop over the genomes, right? So we need to do some looping here as well. For each genome, we collect the 16S gene, right? And then we extract one gene from each genome, put it into a single FASTA file. So we have each genome represented here with one sequence each. And then we do a multiple alignment of this, right? And then we take this multiple alignment and build a tree. And this is exactly what we did in BIN310, actually, more or less. So it's just a way of doing it in R, you could say. And then we get trees of all the different looks. Actually, it's the same data. So let's have a look at the last tree here. And here we have actually colored, because these 36 genomes that we used in this example, these 36 genomes can actually be split into two main groups because they have been assigned a subspecies assignment, right? One is equi subspecies and the other is Suepidemicus subspecies. So we expected actually these genomes to group into two distinct groups. Well, largely they do that actually in this figure. We can see there's one genome here that is colored red, and that means it's this subspecies. I would immediately say when I see this has been wrongly classified as subspecies equi, because all the other ones are here, right? And this one stands out from the rest, and it's sort of mixed in with the other, the blue Suepidemicus here. So I would suspect this one to actually also be a Suepidemicus. But the most important here is that we can see that the differences between the genomes are really, really small, right? And here we can see this sort of length here is a distance of 3%. 3% dissimilarity in the 16S gene. So these are really, really small differences, all of them. And especially here among the equi, it's impossible. They are 100% identical. It's impossible to separate these genomes based on the 16S gene, and almost impossible to separate them from these as well, right? Because they are too similar to each other. There are only some out here who are slightly different. So this picture here actually tells us that the 16S gene is not really suited for separating between the strains within this species, right? And especially within this subspecies here. So this is why we compare based on whole genomes instead of a single gene, right? The whole genomes contain potentially much more information than what you find in one single gene. And also, especially since this 16S gene is too conserved, really. So how do we do these comparisons based on whole genomes? Well, first of all, we can actually do alignment of the whole genomes as well. I write a little bit about this in module 8, but we don't really do any computations along this line. But I mention it. I also mentioned this. We can go looking for this single nucleotide variants, or SNPs, right? like we did in Module 5, a little bit example there at the end of Module 5, and make an alignment of this single nucleotide variants actually instead. So it means that instead of aligning the whole genomes, we only align those positions where they differ, these genomes. At least one genome differ from the rest. So it's two approaches along the same line, you can say, because both of them end up with alignments. And then we have the pangenome approach that we focus a little bit more on this time. And here we use two different approaches to build the trees. Either the pangenome tree based on this pan matrix. I will say more about this shortly. And then a core gene alignment, which is more like the approaches up here, right? But instead of aligning the whole genomes or the SNPs, we align the genes, or at least some of the genes. The other sort of focused approach to comparing the whole genomes is by using these K-Mer based methods. And the big advantage here, I would say, is that you can compare the genomes directly from the reads. You don't need to do all the steps that we need to do before we come to this comparisons here, because there are many steps. So we are independent of all these steps, and that's an advantage in some sense, that each step you need to take introduces some potential errors and uncertainties. So the more directly you can go from your reads to your trees, the better it is in some sense. And it's also super fast. But of course, it's not a perfect method either, because again, this resolution may be too poor with this approach. But that will really depend on a little bit on how you do it, and also on the genomes you are studying. But we will see how we can do these computations as well as these pangenome things. So let me say a few words in more detail about the pangenomes. What is it? Assume you have now many genomes and all of them are from the same species. We know that in principle we can find the genes in each genome. Or we cannot really find the actual genes, but we can predict genes, right? We used Prodigal and Proca, which again uses Prodigal, for this in some previous module. What was it? 6, I think. The only difference is that now we have to do this again and again and again for each genome. And that's why we introduced this array jobs. That's a more technical thing. I will not talk about that now. Let's try to keep the big picture here. But you can surely see that we have to do this again and again and again for each genome, right? So in order to do this in a very fast and efficient way, we use this array jobs. If you do this for a number of eukaryotic genomes, you will find that they all contain the same genes. If you compare, if you sequence the genome of many people, for instance, many persons, and you compare the genes between the persons, you will find they have the same genes. And the same goes for animals and plants and basically all eukaryotes, more or less. There will be a few exceptions, of course, but this is the big picture. For prokaryotes, it's very different, very different. We will see that some genes are found in all the genomes, and these we call the core genes. But we will also see that some genes are shared by many, but not all the genomes, and these are what we call the shell genes. And even some genes are found only in a few genomes, cloud genes. So in eukaryotes, you basically only have core genes. That's what I'm trying to say here. But in the prokaryotic world, we also have a large number of shell genes and cloud genes. the difference between the different strains here is much, much bigger than between the individuals in the in the eukaryotic species. The pangenome is actually the collection of all these genes. All these genes are, if you count all the core genes, the shell genes and cloud genes that you find within a species, then you have the pangenome of that species. Yeah, and the shell plus cloud genes are also sometimes just called accessory genes, right? Because the core genes is something that the genomes must have, but the others are something that they can have, but they don't really have to have them. And as you know, prokaryotes, they share genes across cells, horizontal transfer, as we call it. That's the reason for this happening. So we use this software Rory to do these pangenome computations. And what is actually computed by Rory? Let's have a more detailed look at it. For each genome, we have a GFF file, right? From PROCA or whatever, right? Listing all the genes, or actually the gene sequences, right? In each genome. For example, we have 3 genomes, genome A, B, and C, and we have some gene sequences in each of them. And here it's like around 3,000 in each. And this could be quite typical for a bacterial species, right? 3 genomes from the same species. They have approximately the same number of genes, not exactly the same, but quite similar. And then what we need to do now, and what Rory is doing for us, from all of these 9,000 sequences, because there are 9,000 if you add them all together, we want to group these sequences such that each group is a distinct gene. So it means you have to take each sequence here and compare to each of the 9,000 sequences, and the same for the other 2. Because you can even have within the same genome, you have 2 sequences being from the same gene, right? If it's a repeated gene, right? But especially across the genomes, you will find the same. And notice that they are not identical, right? So for instance, gene number 1 here is actually the same as gene number 10 here, but they are not identically the same. But they are similar enough to be the same gene, right? So once we have grouped all the sequences, then we have the number of pan genes, right? The number of genes in the pangenome, that's the number of groups that we get after this grouping. And we call these groups also gene clusters or gene families. They are just synonyms for the same, these groups. And notice, as I said, the sequences within the same group may be slightly different, but similar enough to be the same gene. And of course, there are some thresholds here that we need to, that will decide how, how dissimilar sequences end up in the same group. So there are some potential uncertainties here, right? When we do this pangenome computation. Are the sequences in the same group really the same gene? Well, depends on the threshold you have used, right? So the major task of this software Rory is to actually find these groups. That's what we do when we use Rory in our modulate. So it means we actually have to compare all sequences to all sequences. Sounds like a big job, and it is, but as I have written in this modulate document, the Rory is doing this actually in a very efficient way. So if we go into the section 5 here, then we see the use of array jobs and so on. when we run PROCA, because we have to run PROCA over and over again many times. And then we do the pan-genome clustering with the row array. Right? And that's really simple once we have all the GFF files from PROCA. The trees we make from these pan-genomes, we make two types of trees. And let's have a look at this first, the tree we make from the pan matrix. And here I have an example where we have actually 3 genomes. So this is the pan matrix, or at least a small part of it. So here we have 3 genomes. In our module 8 examples, we have 36, right? And these have now been grouped. And here I have only 7 of the groups. So usually there are many more, right? Thousands of genes in each genome. So in principle, there is now one group for each gene, right? In this pan genome. One group for each gene in the pan genome. And this matrix contains now either 1 or 0, indicating presence or absence of the genes in the various genomes. So we can see genome A has this gene present. It has also this gene and this gene and this and this, but it's lacking this gene here and this gene here. So we can see that the vector of numbers in the first row here is a sort of presence-absence profile for genome number 1, or genome A. And then you have the similar for genome B here. Present, this one is present, this one is present, this is absent, 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 but this one is present and then absent. And the same for genome C here. So you can see that this pan matrix is filled with zeros and ones. And notice now, group 1 and group 2 are core genes because they are present in all. There are only ones here, right? Group 3 and 4 are shell genes because they are present in some, but not all. Absent in a few. While the last 3 here are typically what we call cloud genes because they are present in one genome and absent in all the others. Present in one of the genomes and absent in all the others. Okay. We compute in module 8, we compute what we call a Manhattan distance between the genomes. So now we want to compare the genomes, right? Compare the genomes to each other based on how similar these presence-absence profiles are for the various genomes. So how do we do that? For each pair of genomes, count how many groups, which is the same as gene, count how many groups they have different values in the pan matrix. Okay, and note that this will mean that the core genes will contribute with nothing here. And let me show you as an example. So here we have a distance table that we need to fill in, right? This should now be filled in with the distance between the genomes. So how do we actually do it? Well, let's compare genome A to B first, the first pair. Then we go through the groups and compare them, or actually count how many groups they have different values, right? So here they have the same value. This is also the same value, so no differences yet. Here we have 1. This is a difference. 1 in genome A, 0 in genome B. The same here. So now we have 2 differences. The same here, 3 differences. And the same here, 4 differences. And here they are equal again. 0 in both. So you only count the cases where we have differences between the genome. So it means that the distance between genome A and genome B in this context is 4. And of course, the this is always symmetrical, so whether you compare A to B or B to A, it's the same, right? And then we start all over again, comparing A to C, for instance. Let me erase this here. A to C. A to C this time. And then we can see again that here there is no difference in group 1. No difference. I type 0 here. In group 2 there is also no difference. In group 3 there is also no difference between A and C. Group 4 is also no difference between A and C. Only here in group 5 we have a difference, 1 and 0. Here is no difference again. And out here in group 7 there is also one difference again. So here the total number of differences is 2, A to C. And so on. And now the last comparison is between B and C, and then you can see again there will be 0 here, 0 here, there will be 1 here. I just type like this, and 1 here, and 1 here, and 1 there. So it's again 4, B and C. And then on the diagonal, it's always zeros, because a genome is always identical to itself. If you compare one of these rows to itself, there will of course be no differences. So here you have the distance table, and we can see right away that A and C is more similar than B is more sort of distant from A and C. So I can almost immediately imagine how the tree will look like. It will be like, yeah, I'm just drawing freehand here now, something like this, right? With A and C more closely related and B a little bit further away. Something like that. But we plug this into a computational engine, you can say, for displaying the tree. So, so this is the comparison of the pan matrix. And notice what these numbers mean. They mean actually how many genes are sort of different in presence-absence status between the genomes. So when we say the distance is 2, it means that there are 2 genes that one of the genomes has that the other doesn't have. So that's the distance we measure now. Count the number of genes where they have a different present-absent status. And notice that the core genes contributes with 0 each time. They will not add any information to this table here because they are present all over. Right? Only these genes out here will have some impact on how the values here differ. And actually, when we if we go to what we did here in module 8, the document, yeah, we computed the pan genome here, and here this figure here we can see the core genes, right? Those who are present in all genomes, all the 36 genomes here. We usually also count those who are present in almost all, because we know that the sequence assembly is uncertain, gene prediction is uncertain, this clustering into these groups is uncertain. So it might actually be that these genes are actually present in all the 36 genomes, but we miss it in one genome for some due to this uncertainty, right? But over here we have the shell genes, those who are present in, yeah, these are present in most genomes, but not all. These are present in fewer of the genomes, but still quite many. And then we have the cloud genes down here who are only present in 1 or perhaps 2 genomes or something like that. These are really rare stuff, and I would say that in many cases these are also false positives from the gene prediction, right? if we have seen that we can get false positives in gene prediction, and they will typically not look like anything else, right? They will not group together with any other sequence because they are rubbish, right? But here is the tree that we make from this, exactly this approach here. And now we can see that, first of all, these two subspecies, they really sort of differ much more now. Right? The red group, the equi gang, is over here, and there's a long sort of distance to the others. Still, this red one here is still appearing to be misclassified. This should never have been an equi subspecies, I think. Seems to belong with the others, the Suepidemicus. But notice that even within the subspecies, we now have some differences. Right? That we saw, we had no differences when we looked at the 16S, but now we see some differences here. But still, there are some of these genomes who have exactly the same genes present and absent. And that's really, this must be more or less exactly the same thing, right? These here. When they don't separate, even at this level, they are more or less identical. Okay, the core gene tree, that's something we also make an example here. So core genes, as we saw, they are found in all the genomes we compare, and they had no impact on the tree we made in the previous slide. But we can also utilize this, because now we can do exactly the same thing as we did with the 16S. Because remember, these genes are actually found in all the genomes we compare. So we can now extract each of these genes from each of the genomes and do a multiple sequence alignment, just as we did for the 16S. And we can, in this way, get many sequence alignments instead of one. And then we just merge these sequence alignments just by placing them next to each other. Because they are all sort of blocks of sequences with the same length now after alignment. So that's simply just putting them next to each other into one very wide multiple sequence alignment. And then we can just make a tree in the conventional way. And this is also something we can do based on the output from Rory, because Rory also computes this for us, at least if we tell Rory to do it, and we do that in the command line we use here. So aligning each core gene. So this tree, if we do it like this tree is based on the core genes only, while the tree in the previous slide was based on all other genes than the core genes. And it could be interesting to compare these two trees and see if they tell the same story. Are the same genomes closely related if we look at them in this way compared to the previous way of looking at them? Because we use two entirely different sets of genes, right? And then finally, the K-Mer-based methods for making trees. And I would say that this is interesting for this reason here, because the approach we did previously require, first of all, we need to assemble into contigs, right? After we have sequenced our genomes. And this is a big job. We have done this, so we know that now. And especially if you have to do this for 100 genomes or perhaps 10,000 genomes, that will be a big job, right? Because each genome takes like half an hour or something. And then you have to go finding the genes. And this is fairly quick though. But we know that this is a step with some uncertainties. And then once you have the coding genes, you need to group them by Rory, right? And this also introduces some uncertainty. Because of this grouping, we have to set some threshold here. How similar should two sequences be in order to be the same gene, etc. So all these three taken together means that the results we get are sort of infested by uncertainties here. It's both time consuming and it adds some uncertainty in each step. So the big advantage of using this mesh software and computing distances from K-Mers is that we can do that directly from the reads. We can actually skip all these 3 steps and go directly from the reads to distances between the genomes. And this is, I would say, the big advantage of this approach here. This is sort of more direct or objective in a sense. So the mesh distance between 2 genomes is computed from the fraction of K-Mers that they share. And I will not talk more about this because that's my video that I linked to in Modulate document, explaining the idea of computing mesh distances, which is actually a very something which is very important to try to understand, I would say, because this way of doing genome analysis is quite new and it has a huge potential, I would say. This has a huge potential because the classical approaches or the pangenome approaches, they will suffer. If it's okay if you have 100 genomes, it's doable if you have 1,000 genomes, but if you have like 1,000,000 genomes, you can give up. But this approach will still work for 1,000,000 genomes. And that's what we will face in the future, I think, quite soon actually. So I will not dive into the details of this one because that's already explained, I would say, in the Modulate document. I should add that these mesh distances, they suffer a little bit from resolution. So if you really have very similar genomes, it's probably not high enough resolution to do it in this sort of quick and dirty way. And then you have to resort to the other methods that we saw earlier. But at least for many applications, this is a good first approach because it's so fast, right? So you can always do this before you do the more cumbersome things, right? Start with this one, and if it gives some resolution, you are happy. But if it doesn't, okay, then you have to do some of the other approaches. But at least then you have tried this out.